Okay, class. So now we're going to start getting into some more specifics with cell signaling. But before we do that, let's just take a quick look at G-protein coupled receptors. These came up in part A of today's lecture. But here they are again. They have a ligand binding domain on the extracellular space, and then they have 7 transmembrane spanning domains. And these actually signal through the G proteins, the G alpha, beta, and gamma. And there's over 700 G protein coupled receptors in humans. And even though it's sort of shown here as a schematic with a ligand binding domain, oftentimes the loop domains between the membrane spanning domains is actually where the ligand binding actually occurs. But this is a very good representation of sort of what they look like. And so they signal through a trimeric G protein, and the trimeric G's proteins have an alpha, a beta, and a gamma subunit. And when it's in its inactive state, the alpha subunit is bound to GDP. And so here's the ribbon diagram of the structure, and here you can see the GDP binding site. And so the G protein coupled receptors often signal through these G proteins, which is how they got their name, G protein coupled receptors, right? And so here's basically what happens when you get activation of the G protein coupled receptor. You'll have, basically it's inactive G protein, and then you'll get the extracellular signaling molecule that will come in, and it'll bind to the G protein coupled receptor, and that can actually activate it by exchanging GDP for GTP, and then you have an activated alpha subunit, and then also an activated beta gamma subunit. And these, in certain instances, these can actually dissociate the alpha from the beta and the gamma. And the alpha oftentimes will go on and activate adenylate cyclase, which can then increase cyclic AMP. And that really sort of one of the big things that it does is activate adenylate cyclase. And here you can see serotonin and what happens with cyclic AMP levels when you add serotonin. Here's just a nerve cell, and you put serotonin on it, and after 20 seconds the red staining here is actually cyclic AMP. So there's a rapid increase in cyclic AMP in response to the neurotransmitter serotonin. Here's how cyclic AMP metabolism works. It starts with an ATP molecule, and then adenylate cyclase will cleave off a pyrophosphate, and then you'll get your sort of cyclic AMP here, and then cyclic AMP phosphodiesterase will hydrolyze this and bring it back to 5' AMP. Okay, guanylyl cyclase basically does the same thing to GTP, as we'll see in a second. But here are some of the cyclic AMP mediated responses. In response to thyroid stimulating hormone, you get thyroid hormone synthesis and secretion, and you can just sort of read these over. In the case with the heart, you'll get adrenaline and increase in heart rate and force of contraction. So just look these over. You don't necessarily have to memorize them. Probably a good thing for the graduate students to know exactly what all of these do, but just in general, the goal here is for you to just appreciate the cyclic AMP mediated responses and know that they're pretty diverse depending on the target tissue that it's occurring in. And so once you have cyclic AMP, what ends up happening is it activates cyclic AMP-dependent kinase, or protein kinase A, or PKA. And so the way that works is you have a regulatory subunit which keeps the catalytic subunit inactive. Okay, and once there's enough cyclic AMP around, that actually will bind to the regulatory subunit and release it, and then you all of a sudden have active pKa, which can then go on and phosphorylate other things downstream. Okay, so this is just an example of allosteric inhibition where the inhibitors actually come and bind, and then you get activation of the regulatory subunit. So here's basically it in a nutshell in a signaling cascade, where you can have basically a signaling molecule bound to a G protein coupled receptor that will cause an exchange of GDP for GTP on the G alpha subunit, which can then activate adenylate cyclase, which will convert ATP to cyclic AMP. The cyclic AMP will bind to the inhibitory subunit on pKa, and that will release pKa and cause it to be activated, and then pKa can enter in through the nuclear pore and phosphorylate Krebs. And then once Krebs is phosphorylated, the Krebs binding protein will associate with it, CBP, and then you can get activated gene transcription. An interesting note for all of you guys here, the CBP protein is actually a histone acetyltransferase. So once it and serves as a co-activator, so it will bind here and then acetylate the histones to help activate gene expression. So in addition to activating adenylate cyclase, the G protein coupled receptors can also signal through phospholipase C beta. Okay. And here's some examples of that where you have basically acetylcholine or vasopressin, and these can elicit a specific response like glycogen breakdown or muscle contraction, depending on where that's happening. Okay. And so before we get into what phospholipase C beta does, we need to look at synthesis of phosphatidylinositol bisphosphate. Okay, so normally you have this lipid phosphatidylinositol. Here's what it looks like. You have the inositol group here connected to a phosphate, and then this kinase called PI kinase for phosphatidylinositol kinase will actually phosphorylate this phospholipid, and then you have another kinase, PIP kinase, which will add another phosphate to it, and these use ATP, and the gamma phosphate is just transferred to create PI45 bisphosphate. Okay, and once you have the PI45 bisphosphate, what phospholipase C does is it actually cleaves this right at the phosphate group here and breaks it down into diacylglycerol and inositol 1,4,5 triphosphate, or IP3. And this activates protein kinase C, and the IP3 actually goes and binds to a receptor on the endoplasmic reticulum to help release calcium. And so here's how that basically works. You can get a signaling molecule to your activated G protein coupled receptor. You'll get exchange of the GDP for GTP on the alpha subunit, and then the beta and the gamma will actually activate phospholipase C, and that will cleave the PI45 bisphosphate into diacylglycerol and IP3, and then IP3 goes and binds to an IP3 gated calcium channel in the endoplasmic reticulum, and that'll release calcium, and then you'll have activated protein kinase C, which is associated with the diacylglycerol, and the calcium will bind to that and cause protein kinase C to become activated. So this is a perfect example of a great signaling cascade that I think you guys should know. For those of you who are headed to medical school or studying for your MCATs, this is a classic example that you'll have to know when you're in medical school, so you might as well take the time to just learn it now. And if we want to see what happens with calcium release in a fertilized egg. So here the egg is getting fertilized here, and you can start to see the calcium released over time, and there's a huge amount of calcium here. And so one of the things that has to be done is, in order for this to work, is the calcium within the cytosol actually has to be really, really low. And so the molar concentration of calcium in the cytosol is about 10 to the minus 7th molar. And it's done that by a couple of different mechanisms. Here are two. You actually have a sodium-driven calcium exchanger. So sodium flows in and calcium is pumped out. And then you also have an ATP-dependent calcium pump that actually uses the hydrolysis of ATP to pump calcium out of the cell. Okay, and this makes sure that the cytosolic calcium levels are actually low. You also have a pump in the endoplasmic reticulum where you have an ATP-driven calcium pump that's pumping calcium into the ER, and then there's also molecules that sequester calcium in the cytosol, and there's also a proton exchanger, a symporter here, that's pumping calcium in. And we'll learn more about symporters and antiporters later on in the term, but suffice it to say for this, there's all these different mechanisms that keep calcium low and store it in different intercellular compartments, specifically the endoplasmic reticulum, because that's where most of the calcium is stored, and that's where the IP3 gated calcium channel is actually located, in the endoplasmic reticulum. And so here's the vasopressin response, and it changes depending on the concentration of vasopressin. And if you just sort of follow the spikes in calcium release here, you can see as you increase the vasopressin concentration, you're actually increasing the release of calcium. Okay. So calcium can do a couple of things in the cell. One of the things it also does is it can bind to calmodulin. And so here's the calmodulin molecule. You have calcium binding sites on it, and then once it's activated, or once the calcium's bound, it can help activate CAMP-Kinase II. Okay, so this is just an example of the conformational change that it'll undergo. But here's CAMP-Kinase II. So typically you have calcium-independent activity of CAMP-kinase, and eventually a protein phosphatase will make it inactive, and then you have your calmodulin protein, and once calcium is released, that will bind to the calmodulin, so you have calcium calmodulin, and that will bind to the inhibitory domain on CAMP-Kinase II, causing it to become activated, and then you'll basically get autophosphorylation, where the gamma phosphate of ATP is actually transferred to the protein, and then your CAMK2 is fully active. Okay, so this is sort of simplified. There's actually 12 subunits in CAMP-kinase, but here's how calcium calmodulin helps to regulate CAMP-kinase activity. And you can see that sort of here with low spikes, you don't really get any calcium calmodulin. You actually need high frequency calcium release, and that will actually lead to a gated sort of dose response here in CAMP-kinase activity. Okay, so if you're releasing only every 20 seconds, pretty much nothing changes. The calcium's just pumped back into the endoplasmic reticulum and out of the cell. However, if you're having high-frequency calcium oscillations, that can bind to calmodulin and increase calcium calmodulin kinase 2 activity. Now, G-protein coupled receptors and adenylate cyclase are also important in sensing, okay? And so if in your sort of nasal passageway you have olfactory neurons, and here you can just see a nice scanning EM of one, and so upon binding, they can activate adenylate cyclase, which then can stimulate the formation of cyclic AMP, and the cyclic AMP then binds to cyclic AMP gated ion channels, allowing a influx of sodium that can depolarize these olfactory neurons and then lead to a thing. So lead to the sense of smell. And so this is basically how, or allows your brain to interpret the sense of smell. And so this is actually how we smell things. We basically use G-protein coupled receptors and adenylate cyclase, which then binds to a cyclic AMP gated ion channel, which causes depolarization of the olfactory neurons. Okay, and then the axon goes off to our brain. Same kind of thing happens with vision, except we use cyclic GMP. Okay, and cyclic GMP is synthesized by guanylyl cyclase, and so here you have your guanine, your ribose, and then your cyclic phosphate here. And so this actually works in the rod photoreceptors in our eyes, and it's for basically non-color vision in dim light. Okay, so here is basically a photoreceptor cell. We have discs of photoreactive membranes here, and then you have your your inner segment, your cell body with the nucleus, and then your synaptic region down here. And the rod photoreceptors up here in the photoreceptor membrane actually use a molecule called rhodopsin, which is a G-protein coupled receptor that actually doesn't have any chemical ligand. It actually uses a photon of light as its sort of signal. Okay, and once a photon of light is absorbed, the cyclic or GTP is converted to cyclic GMP, and that actually binds to cyclic GMP-gated cation channels. To give you guys a better idea of how that works, so in the dark, the cells are actually depolarized and the cation channels are open, and here is your active, your inactive rhodopsin here. When you start to see low levels of light, the rhodopsin becomes activated. That converts GTP to cyclic GMP, which then will bind to these cation channels and cause them to close. Okay, and then the cell becomes hyperpolarized. And so in sort of the dark, you're getting sort of a steady release of neurotransmitter. However, when the cell becomes hyperpolarized, the neurotransmitter vesicles, or the vesicles containing neurotransmitter, don't fuse. Okay, and this is how we are able to sort of see in dim light. So it turns out there's a whole family of trimeric G-proteins, and here's just some of the examples of them. You don't have to necessarily memorize them. The graduate students, you will come across these throughout your career, so it's probably a good idea to learn them now. And the point being that, different G-protein coupled receptors will actually interact with different G-proteins and do specific functions. Okay, and so getting back to light transduction, you'll have one photon of light sensed by one molecule of rhodopsin. This will actually cause 500 G-proteins to become activated. Okay, and those 500 G-proteins that are activated will then activate guanylyl cyclase, and 500 cyclic GMP molecules are activated, and those GMP molecules are hydrolyzed, and they cause the cations to close, and ultimately what you get is sodium ions are prevented from entering the cell for a short period of time, and then there's a change in the membrane potential and the signal is relayed to the brain. Now in this whole process with G-protein coupled receptors, they can become desensitized, right? And we looked at a little bit of an example of desensitization at the end of part A. Okay, but once you have your activated G-protein coupled receptor, that can actually stimulate GRK, or the GPCR kinase, KIRK, to phosphorylate the G protein. And then in certain instances, this protein, arrestin, will actually bind to the G protein coupled receptor. Now, in the fly eye, when it's actually, when you have your rhodopsin here as your G protein coupled receptor, when arrestin has bound to it and it's been phosphorylated by the GERK here, arrestin will bind, and it actually also then gets endocytosed. Okay, so once again, there's another sort of mechanism of desensitization, and a lot of it's mediated by the G protein coupled receptor kinase and arrestin binding. Okay, now on to receptor tyrosine kinases. Okay, so these are another family of receptors that cells use. There's a whole bunch of them. The EGF receptor, it has a cysteine-rich domain, which binds the ligand, and then a tyrosine kinase domain. So this is epidermal growth factor. You have the insulin receptor, the NGF receptor, the platelet-derived growth factor. These have immunoglobulin-like domains. The fibroblast growth factor receptor, VEGF, which is somewhat vascular endothelial growth factor receptor, and also the EPH receptor for the epinephrine receptor there. And so here are just examples of some of them. And it's afferrin receptors, I'm sorry. So here they are, the different ones. You guys should know a bunch of these. One that I think should really jump out at a lot of people is this VEGF receptor. So oftentimes cancer cells will secrete vascular endothelial growth factor, which actually stimulates angiogenesis. And so things that block the VEGF receptor have been of high interest to a lot of pharmaceutical companies, because the idea is if you could sort of starve a tumor of oxygen by sort of getting rid of angiogenesis and blood flow to that tumor, it will help to kill the tumor. So that's just one of that's very interested, or pharmaceutical industries are very interested in. Another one is insulin growth factor receptor. So you guys should know some of these ones. I won't ask you to memorize them, but I will potentially use them as an example on a test and tell you what they are and what they do. But here's how RTK activation works, the receptor tyrosine kinases. You have your inactive RTKs with a tyrosine kinase domain, and then you'll get a signal molecule, and that can actually bring them together, and then they end up cross-phosphorylating each other in sort of a transphosphorylation reaction. Okay, so the signaling molecule actually brings them in close proximity so that they can then cross-phosphorylate. And because of that cross-phosphorylation, you can actually get dominant negative mutations in them. Okay, so here is basically a normal tyrosine kinase. If you have a mutant tyrosine kinase along with that, then all of a sudden when the receptor molecule binds, there can be no cross-phosphorylation. So these dominant negative mutants in receptor tyrosine kinases have been very useful in sort of studying them and understanding what they all do in various signaling cascades. And what's great about it is you can sort of ectopically express these in just a mammalian cell culture to sort of figure out what they're doing, right? Because they're dominant negative mutations. So the RTKs also have a whole bunch of accessory factors. Okay, so once they've sort of transphosphorylated themselves and are activated, these phosphotyrosines often serve as docking sites to recruit other accessory factors to them, and then they can go on to signal other proteins. And here's sort of a better example of this. This is the platelet-derived growth factor receptor. So when the platelet-derived growth factor has bound to the receptor, you'll get transphosphorylation. And there's all of these tyrosine residues where the phosphorylation occurs, and different subunits will bind here. You have a PI3 kinase, which will bind, and then a GTP activating protein, or a GTPase activating protein, or a GAP, as well as phospholipase C. And what allows them to interact with these phosphotyrosines are these SH2 domains. Okay, so the SH2 domains will interact with the phosphotyrosines. And here's basically what an SH2 domain looks like. And in one instance, you're going to have basically the phosphotyrosine that will interact with that the SH2 domain interacts with, and then you'll have an adjacent amino acid which sort of specifies where it's what SH2 domain will bind there. And so to give you sort of a better example of that versus this sort of ribbon diagram of the SH2 domain, you can sort of almost look at it like this, where you have sort of a site for the phosphotyrosine where that's going to bind, and that's going to be pretty universal in all SH2 domains. But then you're going to have a specific sort of site for a specific amino acid which will be adjacent to it on the phosphotyrosine. So if we go back, you can see the phosphotyrosine at 740 might have an adjacent amino acid nearby that the SH2 domain associates with that's different from the one that occurs at amino acid 771. And so that's why the PI3 kinase binds up here, and the GAP protein will bind down here. So it's just the specificity that's laid out in this sort of second amino acid binding site. So the phosphotyrosine is pretty universal, and this site is actually what's different. Okay, now on to monomeric G-proteins. And I call the G-proteins with the G-protein coupled receptors trimeric to sort of distinguish them from these other G-proteins. And we've already talked about some of these through class. We have the RAS proteins. There's HRAS, KRAS, and NRAS. And these relay signals from RTKs. Some other ones we'll look at later on in the term will be the RAB proteins, ARFs, and RAN. And so RAN is actually involved in nuclear import. And then there's some other ones, the Rhos, which we'll see, the Rho family, which we'll see in just a little bit. And they're oftentimes help to change the cytoskeleton. Okay, but let's look specifically at the RAS family and sort of how they work. And to do that, there was a great experiment done in Drosophila, okay, to look at how RAS is involved in sort of signaling, okay, and it had to do with eye development. Okay, so this is the compound eye of Drosophila, and it's been sort of a great model system to study the monomeric G-proteins and the receptor tyrosine kinases that signal through them. And some of the proteins and genes that have come out of this were SEV, BOS, and SOS. SEV is sevenless, BOS is bride of sevenless, and SOS here is son of sevenless. And we'll see how those work. But if you look in this sort of compound eye, each one of these little islets here is what's considered an ommatidia. And the ommatidia in a fly eye actually has 8 cells. Okay, so the 8 cells make up a single ommatidium in the compound eye, and the development sort of follows this schematic up here. Okay, the R8 cell develops first, and then that's followed by development of the R2 and the R5 cell, and then R3 and R4 cells develop, and then R6 and R1 develop, and the last cell to develop is R7. And so if we look here, you can sort of see one single ommatidium here, and you can see the various cells here. Here is R8, R1, 2, 3, 4, 5, 6, 7. And so if we go back, the 8 cells that we're talking about is in one single ommatidia here, okay? And so they actually used this sort of developmental pathway here to actually screen for regulators of development. And, you know, one of the first mutants that they found was this strain of flies called 7-less, where there was a mutation that basically caused no R7 cells to develop, okay? And so after sort of teasing apart the whole pathway, they found all of these different proteins that are involved here. So it turns out that 7-less is a receptor tyrosine kinase, and it associates with bride of 7-less. Okay, and so once bride of 7-less, and the bride of 7-less is actually on the R8 cell, and so when the R7 cell is in contact with the R8 cell through this interaction between bride of 7-less and 7-less, you get phosphorylation of a tyrosine through the receptor tyrosine kinase, and that recruits the adapter protein DRK binding via the SH2 domain, and then you have this protein called SOS, or son of 7-less, binding to the SH3 domains. So it turns out son of 7-less is a RAS-GEF. And so if you go back to part A of today's lecture, you'll remember that the GEF is a GTP exchange factor. So it activates the exchange of GDP for GTP on a small G protein like RAS. So what son of 7-less is doing here is it's activating the exchange of GDP to GTP on RAS, and then RAS becomes activated, and then it can lead to downstream signals such as the MAPK kinase cascade and other things. So it's really important that you guys appreciate that son of 7-less is a RAS-GEF and how this whole process works. In years past, I haven't held everybody to sort of memorize this, but this year I think you guys did so well on the first exam. I think this is you know, a signaling cascade that I think I want to hold you guys to, just because it's so important, right? Because, you know, RAS is sort of locked in its GTP bound form in so many cancers, and I just think it's such an important thing to understand that son of 7-less is a RAS-GEF, and it causes the exchange of GDP for GTP. And just in general, the whole system and the screening for bride of 7-less and 7-less and all of this is such a great story. Okay. So you can sort of follow RAS activation using this technique called FRET, for fluorescence resonance energy transfer. And the way FRET works is you shine well, first of all, you have to fuse RAS to a protein like YFP, yellow fluorescent protein, and you can excite that protein with one wavelength of light, like 476 nanometers, and then that will emit at a different wavelength of light at 528. And then you can sort of measure when it's activated if you have a fluorescent red dye attached to GTP, and when you get the exchange of GDP for GTP, the light energy emitting from the YFP can be absorbed by the fluorescent dye, and it will actually emit at a different wavelength of light. Okay, so what ends up happening is you're just shining one wavelength of light, you're activating this molecule, and then rather than just emitting, if there's another molecule nearby that can absorb it, it'll emit at a different wavelength, and you can then sort of measure the different wavelengths of light here. And that's how FRET is used, and it's a very common technique to understand protein associations within live cells, because you can just fuse YFP and RFP onto different proteins. And so what you'll end up getting is basically you can sort of measure the number of activated RAS molecules. If you add epidermal growth factor, for example, you can see the amount of activated RAS molecules using this. Okay, so it's just a convenient way to measure it. You guys don't have to spend a lot of time on this. Graduate students, you will come across FRET quite regularly in a lot of the literature that you're reading. But once RAS is active, it actually goes and signals onto the MAP kinase cascade. Okay, so what you end up getting is RAS to RAF, which then signals to MEK, which then signals to ERK, and then those can go on and phosphorylate other things. So this MAP kinase cascade, I'm sure you guys have all seen it at some point in time. RAF is a MAP kinase. So once RAS activates it, the MAP kinase becomes active, and that phosphorylates the MAP kinase, and then the MAP kinase phosphorylates the MAP kinase, and then the MAP kinase can go on to phosphorylate proteins that can either elicit a change in activity or it can change gene expression. To keep this all sort of well and organized, you actually have scaffold proteins. So if you guys remember back to the first part of the lecture when I was talking about the SHMU and the yeast mating, what you end up having is the yeast mating hormone. It'll bind to a G protein coupled receptor. It'll eventually activate through RAS, and then RAS will activate the MAP kinase, then to the MAP kinase, and then finally to the MAP kinase, and then you get the mating response and actually the formation of the mating projection and the formation of the SHMU. Okay, same thing happens with osmolarity. You can have an osmolarity sensor, and then that'll have a scaffold protein, and you'll get signaling through the various MAP kinase, the MAP kinase, and then the MAP kinase. Okay, turns out the scaffold protein in yeast, which I remember from when I was in graduate school, is actually called sterol 20. And if you get rid of the scaffold, this gets all messed up, and the yeast are actually sterile, which is how it got its name. Okay. Okay, another small G protein is called Rho. It functions sort of a similar way, where you get the GDP bound form of Rho inactive, and when it's the GDP is exchanged for GTP, it becomes active, and this is very important in axon growth cone formation. And so here's how that sort of works. You have your Epherin A1 on an adjacent cell, and the growth cone will grow. And if there's a, if the growth cone doesn't want to be associated with this cell, you'll get activated the EPH4 receptor, which then will lead to activation of a tyrosine kinase, and then activation of a Rho GEF, which will cause the exchange of GDP for GTP, and this will actually lead to activated Rho, which causes myosin-mediated actin filament contraction and growth cone collapse. Okay, so if the growth cone doesn't want to be there, it will actually collapse through this signaling. And so normally the growth cone is extension. You have an inactive one, and the Rho GEF is actually not active, and so the growth cone is sort of extending without binding, and then once there's binding, what will end up happening is the Rho GEF becomes activated, and Rho A the GDP is exchanged for GDP on Rho A, and you get growth cone collapse. Okay, now on to PI3 kinases, and we've already sort of seen these before, the phosphatidylinositol, but the phosphates actually get added by PI3 kinase. Okay, so here you have phosphatidylinositol, and with the PI3 kinase, that will become PI3 phosphate, and then if you have PI4 phosphate, PI3 kinase will also add a kinase phosphate onto the 3-carbon, and also you can have PI4,5 bisphosphate, which actually can get cleaved into phospholipase C and IP3, but also it can become PI345 triphosphate. Okay, and PI3 kinase adds the phosphate to the number 3 carbon on the inositol ring here. And so that becomes sort of important in signaling for apoptosis, okay, or survival signals. Okay, so you'll have a survival signal, and this inhibits apoptosis. So you'll get a survival signal bound to a receptor tyrosine kinase. These will cross-phosphorylate themselves, and then through an SH2 domain, you can get an activated PI3 kinase, which will convert the PI4,5 bisphosphate to PI345 phosphate, and then these proteins PDK1 and AKT will actually bind to that, and then you get activation of AKT through mTOR and the PDK1. That will cause a dissociation, and then you have your inactive apoptosis inhibitory protein, BAD, and that will phosphorylate it, and the inactivated BAD binds to the 14,3,3 protein, and then your apoptosis inhibitory protein will bind here. So we'll revisit this when we're actually covering apoptosis, but I thought it would be good to sort of introduce it here. Now, one of the things that you guys really have to appreciate, and I sort of mentioned this in part A of today's lecture, is a lot of these signaling cascades, they're interconnected, and they have a lot of crosstalk. And so we saw in sort of the classical sort of signaling cascade where it can integrate signals and it can also spread signals. And so rarely do these things exist in a vacuum. Okay, oftentimes you'll have G-protein coupled receptors and receptor tyrosine kinases sort of working together when they're getting signaling molecules. And we sort of saw that with the schematic where multiple inputs are being put into a cell, and it can tell it to basically just stay the same and grow and survive. It can tell it to divide, it can tell it to differentiate, or it can tell it to sort of undergo apoptosis. And we saw an example of things that prevent apoptosis from happening by getting these sort of survival signals. But this figure right here sort of really just goes to show you how complicated things can get. And so, there's no need to necessarily memorize this, but sort of appreciate all of the different things that can happen with, either signaling through a G-protein coupled receptor or through a receptor tyrosine kinase. So the G-protein coupled receptor is going to activate G-proteins, and the G-proteins can go on to activate adenylate cyclase, leading to cyclic-AMP and pKa activation, or you can get phospholipase C, cleavage of IP3, calcium release, which then can bind to calmodulin and modulate CAMP-kinase activity. And then you'll also have diacylglycerol, which then can affect PKC activity. And then you can have basically the receptor tyrosine kinases activating PI3 kinases and signaling through the RAS MAP kinase cascades, and ultimately there can be tons of crosstalk. And I just want you to sort of consider all of this in the context of all of the different G-protein coupled receptors and receptor tyrosine kinases that exist. And really sort of just take this in from a 10,000-foot view. I don't necessarily require students to memorize this, but just appreciate all of the different integration events that can happen here. Okay, and so here's basically what a receptor tyrosine kinase looks like. On the outside of a cell, here's the plasma membrane down here, and this is actually the human growth hormone receptor with human growth hormone shown here in orange and binding to it. Another signaling cascade that's really sort of important, and I think you guys should probably learn this, it's simple enough to know, is the Jak-STAT signaling cascade. Okay, so Jak is the Janus kinase, and STAT is signal transducer and activators of transcription. Okay, so these tend to work with cytokines. And if you remember in sort of the Part A of today's lecture, I talked about the cytokine storm that can happen with COVID-19. And so what happens here is you basically get, you have your cytokine receptor with the Janus kinase here, Jak, bound to it. And then the cytokine will bring the cytokine receptors together, and then you'll get cross-phosphorylation of Jak, which then will phosphorylate the cytokine receptor, and then that will recruit STAT, and then the Jak can actually phosphorylate STAT, and then once the STAT is phosphorylated, it becomes active, and then it enters the nucleus, and other gene regulatory proteins can bind, and then it binds to a cytokine response element in a target gene. Okay, and the reason I think I want you guys to know this time around is because it turns out that it's really cytokines in general are super important in a lot of diseases, specifically autoimmune diseases. And some, examples of autoimmune diseases where the cytokines are misregulated are, arthritis, multiple sclerosis. There's just a whole bunch of them. Crohn's disease, the list is quite large. And oftentimes the cytokines are just high, and there's high levels of intercellular TNF-alpha or the interleukins. Interestingly, in multiple sclerosis, you have an increase in this specific T helper cell population called Th17 cells, and they secrete IL-17, which can actually cause the breakdown of muscle and other things where your immune system is attacking the muscle cells. And so one of the great things with these cytokines, though, is oftentimes when they're sort of discovered what they do, it's easy to generate a human monoclonal antibody to sort of inhibit the activity of these. So in the case with TNF-alpha, there's a number of approved human monoclonal antibodies. Probably one of the best known and actually the best-selling drug that's ever been made is a drug called Humira. I'm sure many of you guys have seen TV commercials. It's a human monoclonal antibody that recognizes TNF-alpha. And there's actually a couple of IL-17 monoclonal antibodies that I know about that are actually being, at least in clinical trials, for treatment of multiple sclerosis and other autoimmune diseases. So here's just an example of some of the signaling proteins and how they function through the Jak-STAT signaling cascade. No need to necessarily memorize these, but just remember the cytokines are really important in immunity and are often misregulated in autoimmune diseases. And just go back and sort of look this over. It's simple enough that I think you guys can sort of catch on to how it works, right? But this is a great mechanism for cytokine signaling. Okay. Okay, another really important signaling cascade is called TGF-beta signaling. Okay, and so you have TGF-beta, and it actually binds to the TGF-beta receptor, which is has a serine-threonine kinase domain, and you have two different TGF-beta receptors, a type 1 and a type 2. They'll come together and you have cross-phosphorylation, and then that will lead to phosphorylation of this protein called SMAD. Phosphorylated SMAD 1 or SMAD 2 or 3 actually associates with SMAD 4, and these can then enter the nucleus and bind to TGF-beta response elements in the genome and lead to target gene transcription. Okay. So here's just an example of the different kinases. You have your receptor tyrosine kinases like the insulin receptor and the platelet-derived growth factor receptor and the epidermal growth factor receptor. And then you have other tyrosine kinases like SARC and Jak. And then you have your serine threonine kinases like cyclic-AMP-dependent kinase and protein kinase C, MAP kinases, and the TGF-beta receptor and whatnot. So that's pretty much all I have for today. For Thursday, we're going to cover the cytoskeleton. So if you've fallen behind on your reading, now would be a really great time to catch up. And I'll look forward to talking with all of you later this afternoon. Okay? Bye-bye.